Let's reflect on Chapter 4 in 5 minutes or less. So if we want to collect data, we're going to take a sample or a census. So we can either give a survey or an observational study where we would just sit like at a table somewhere and just observe people as they walk by, or we could actually do an experiment where we would have a treatment that we would put on the subjects. So let's focus on surveys. So 2 types are convenience and voluntary surveys, but these are biased in nature. They're easy, but they have negatives attached to them. Simple random sample, stratified, and cluster are all ways to randomize your sample group for your survey. If you're looking at a stratified group, you're going to want homogeneous groups. That means they all have a similar trait, and then you take a simple random survey from each group. Cluster would be heterogeneous, and you'd take one cluster and you would survey everyone in that cluster. Now be careful when you're taking a survey. You want to eliminate as much bias as possible. Some types of bias we've talked about are response bias. That's where you're going to change your answer. You're not going to tell the truth because of who's asking it or how the question's asked, the wording of the question. And then there's non-response, where you just don't answer the survey at all. In any survey, you want to eliminate as much bias as possible. Randomizing helps with this. But if bias is present, it's a systematic flaw, and it will result in a sample that doesn't represent the population. Now, bias is very different than sampling variability. Sampling variability happens naturally. You take one sample of 5 randomly selected men and you find their average height. And then you take another sample of 5 randomly selected men. You expect them to have a different average height. That's sampling variability, also known as sampling error. It occurs naturally. It is not biased. It can't be avoided. All right, moving right along to experiments. In any experiment, you need control, replication, and randomization. Now, control doesn't mean you have a control variable. It means you're going to try to control as many lurking variables as possible. Replication, you're going to repeat the experiment several times. And randomization, probably one of the most important things. You must randomly assign subjects to treatment. All right, so ways of randomizing. Well, if there are variables you want to try to control, you could do a block design. This is where you would split your sample group by some sort of lurking variable or some sort of common trait, such as gender. You could also do a matched pairs design, and that's where each subject in your experiment is going to get all the treatments in that experiment. So they do the test once, maybe in a placebo, and then again with the experiment. Finally, how do we know, or how can we tell what conclusions we can make? So we have to examine, were individuals randomly selected, and were individuals randomly assigned to a treatment? So we have to answer these questions, and then we can decide what kind of conclusion we make. So if yes, individuals were randomly selected and randomly assigned treatment, then we can make inference about the population and inference about cause and effect. Take a look at this chart a little bit further in detail to see what other conclusions can be drawn based on whether or not individuals were randomly selected and whether or not treatments were randomly assigned.